hear me okay? Is it picking it up? Am I loud enough? Okay, awesome. Good afternoon, everyone. I am Lydia Fitzgerald, and I am a second-year master's student. I am working with Dr. Edwin Ritchie, and my research is focused on broiler litter and looking at different nitrogen mineralization rates between the bedding types used, and then also between the application methods. So poultry is a big industry in Kentucky. It's actually the #1 agricultural commodity here in Kentucky, and we rank 7th in the nation in broiler production. So that totals up to 3,000 houses producing over 450 tons of broiler litter every year. So since it is widely available for producers to use, it makes it a very valuable and economical source of nutrients. There are many benefits to using broiler litter in crop production systems. First of all, it's a very high value manure, which just means it has a very dense nutrient content. And so when you apply it to crop production fields, it actually increases the soil organic matter. And this in and of itself has many big benefits for the soil. It improves the soil structure, infiltration rates, makes your soil better able to hold nutrients and water. But in addition to this, when crop producers are using this as opposed to synthetic fertilizers, they won't necessarily see a yield cut, which is a big deal because we want them to be able to use it without seeing decreases in yield. There are some concerns though with broiler litter. Since it is such a nutrient-dense manure, this causes issues with leaching of nitrate into groundwater, excess nitrogen and phosphorus. In fields where it's applied, can be issues with runoff. And then also when producers are thinking about applying it for nitrogen, a lot of times they may forget to account for the other nutrients that they're applying, such as phosphorus and potassium. So this can lead to excess levels of those in these systems. So nitrogen is very important in cropping systems, as I'm sure we all know, but a lot of times it's in the organic forms, which aren't plant available. And so nitrogen mineralization is important because we want to take it from those organic forms and put it into inorganic forms of nitrate and ammonium. And so that's what we're looking at in this study, is to look at the nitrate and ammonium and see when it becomes available from the broiler litter that we're applying. The objectives of my study are to, first of all, look at nitrogen mineralization rates in terms of the different bedding materials used in poultry production, so specifically rice hulls and then wood-based beddings. And the second objective is to look at differences between incorporation practices. So that's whether it's just applied on the surface or whether it's well incorporated into the soil. There are many factors that impact the nitrogen mineralization rate, and I'll just be going over a couple of them because there are many. But a lot of times it has to do with, obviously, the litter composition, so what's in the litter. And this is impacted by the in-house practices. So this can be things such as the stockpiling practices, how they compost their litter, even the type of poultry that are in there. So all of these things are very important when looking at the composition of the broiler litter itself. The bedding material is important because in the Midwest, they tend to use rice hulls as the bedding material. So if you get a litter from Western Kentucky, a lot of times it's going to have rice hulls. But if you go over to the East Coast or the Southeast, or in some areas in Kentucky, it'll have wood-based beddings. So this is primarily just based on what's economical and what's available to the producers to use. And then the application method has an impact, and this has a lot to do with whether you're in a no-till system or not. So if you're in a no-till system, producers are likely just going to be broadcast applying on the surface, whereas if you're in a tilled system, they're going to be incorporating it into the soil, and this has an impact on where the nutrients go and what happens to that litter. So some of the other factors that are important to keep in mind are ammonia volatilization. This is especially prominent and a big potential for it in poultry litter because of the high amount of urea in poultry litter. And so the particle size is important, the carbon to nitrogen ratio, pH and temperature all impact the form of nitrogen that's in the soil and in the litter, and then also the microbial activity in the soil, which influences the mineralization rates. So the first study I conducted looked at the application method and then also at the rate. So this is a laboratory incubation done out at Spindletop in incubation chambers, and we utilized destructive sampling at various sampling dates between day 1 and day 74. So we applied broiler litter, and then we also applied an ammonium nitrate control at a rate of 100 pounds of nitrogen per acre, and then also at a rate of 400 pounds of nitrogen per acre. so 1x and a 4x rate. And then half of them, we incorporated a mixed well into the cups. And the other half, we just spread evenly on top and then incubated them. And so we also included a soil-only control, and we had 4 replications that were in 4 different blocks, 4 different incubators that we conducted the study in. And we used 1 litter for this study. We just used one of the common litters found in Kentucky that was rice hull bedding, and it was from a full clean out. The second study is set up very similarly, except for we were looking at the bedding material. So we currently have 2 rice hull litters, and then we have 5 wood-based broiler litter samples. So this will be set the same way with 4 replications, 4 blocks, and we will be incorporating all of these, mixing them in really well, and applying them at a rate of 100 pounds of nitrogen per acre. We'll also be doing the same destructive sampling, and we'll have a soil control and then a nitrogen fertilizer control. So for the incubations, they're incubated at 25 degrees Celsius and 85% humidity, and our goal is to keep the moisture content at 50% of field capacity. And so these were just trying to keep it, optimal for microbial growth, for microbes to be able to, mineralize the nitrogen. And we used 150 grams of dry soil and watered biweekly. For the broiler litter that we did collect, we sent out a survey to the producers and tried to get as much information as we could from them about their in-house practices, and just to get an idea of what was going on in the houses that impacted what the litter was like. So we also did a routine analysis on the litter to get preliminary information about the litter before we started the study. The soil that we used was a Kreider silt loam, and we pre-incubated it for about 2 weeks at 25 degrees Celsius just to stabilize microbial populations. And then we also analyzed this just to get preliminary information before starting the study. When we sampled at the specified sampling dates, we'd remove the samples, get the moisture content, process them, and then immediately do a KCl extraction before much happened, with the nitrogen and the forms of nitrogen, to try to get as accurate of a picture as possible as to what was actually happening inside of the cups. So we did a KCl extraction and analyzed them for inorganic nitrogen on the latchet. And we also are doing a water extraction to analyze for orthophosphate, and then a malic-3 extraction to analyze for phosphorus, iron, and aluminum. We are getting the pH data and also sending them off for carbon and nitrogen analysis. So this table is showing you what information we got from the actual producers. So the important things to notice here are that we have 4 litters that are from Western Kentucky that we collected out there. And then we actually have 3 that are from Delaware. We had some faculty out at the University of Delaware that were willing to help us collect litter from out there. The next thing to notice is that we had 2 rice hulls that were both from Western Kentucky, and then the rest were wood-based bedding materials. And of the wood-based ones, they had various sizes, and so we also have to take that into account. But besides that, this is just to show the variety of in-house practices. And then this table is a lot, but it's to show that there were differences in the actual litter composition. So this is showing the preliminary analysis that we did of the actual litters. There's 2 main takeaways from this table. The first one is to look at the carbon to nitrogen ratio of the litter samples. So you see the first 2, those are both the rice hull litters, and they have a much lower carbon to nitrogen ratio as compared to the 5 that were wood-based. And that's as you would expect, just from knowing the differences in the materials, and then also from what I have read in literature. That's the common theme, is that they do tend to have a lower carbon to nitrogen ratio. The next thing I wanted to look at were litters 3 and 4 from Western Kentucky, and that's because they came out of the same production house. So both of those came from the same house. The only difference is litter 3 was composted and litter 4 was taken directly from the poultry house. So I just wanted to point that out to show you how much of a difference there is when you do different composting practices, and how differently the litter composition looks when you do that. So the results that I'm going to be sharing with you today are the inorganic nitrogen results that I got from the latchet, and those were for sample dates 1 to 28. So this graph is showing the amount of ammonium in the sample cups over 28 days. And this graph is just showing the 4 poultry litter treatments, not the ammonium nitrate in the control, just the poultry litter. And so there's high rate, and there's low rate of poultry litter, and then there's tilled and no-till samples. So in the green, you'll see the tilled samples, and in the blue, you'll see the no-tilled samples. And so if you're looking at this, you can see that after day 14, there was a sharp decline in the high rate samples. And if you look at the next chart, you can see where the nitrate and nitrite results show an increase after day 14. And so, you know, needless to say, it looks like there wasn't necessarily more nitrogen being put from organic to inorganic from nitrogen mineralization, but rather just changing forms. And so you can see that on this last graph, which is inorganic nitrogen for all of the poultry litter treatments. So if you have seen graphs from incubation studies before, you'll recognize that there's not much going on here. You would typically want to see a curve in this upward, so you can see where nitrogen is being mineralized from the organic pool to the inorganic pool. And so obviously there is something going on here. There is a limiting factor, and it was very evident what it was. So it was water. Fairly simple. So the water content, we were trying to keep at around 15 to 17% gravimetric water content. And so you can see that it decreased very sharply and went down to almost 4%, and that is not enough water for microbes to be happy. And so because of that, we saw not much nitrogen mineralization happening over the first week or two of this study. So for the following studies that we're going to be doing, the bedding material study, and then also a follow-up study to this one with the higher moisture content, we're planning on watering more often, multiple times a week, and then also maybe changing the actual layout of the cups to try to keep the moisture content up in these studies. And once we do that, it should help some nitrogen mineralization to start happening in the cups. So the outcomes of the studies, once they're completed, are to help refine the nitrogen mineralization standards to hopefully include bedding materials and application methods, and then to also produce a publication on the effect of litter bedding on nitrogen mineralization rates. We'd also like to make updates to the economics of management decision-making tool that exists that producers use to help them make decisions about applying broiler litter and whatnot. So those are the three outcomes from this. I'd like to say thank you to my committee and to everyone else who has been very helpful in helping me get the study started and completed. So I can now take any questions that anyone has.
Thank you, Lydia. Does anyone have any questions for Lydia?
So what percent of the water holding capacity was your starting moisture content?
What percent of the water holding capacity?
Yeah.
We were going for 50%. Yeah, and it was about 17% gravimetric water content. And so we were gonna probably also increase that, like increase the starting out water content to help it. Yes. Well, I'm actually working on that right now and trying to figure out which one was the best. And I was hoping to have it finished for this, but I did not. So I'm actually not sure yet which one would be the best one to use, just because I'm still looking at them and trying to figure out what would be best, working with a couple of the statisticians to figure that out. Yes.
That's curious. Do you think there could be some nitrification going on? You saw the increase in the
Yes, I think there could be some ammonia volatilization happening inside the incubation chambers, but also nitrification happening. That would be, when I've looked at it and kind of analyzed a little closer, that's what I figured was happening in the cups, was that it was, you know, changing form.
Thank you, Lydia. Our next speaker is Ryan Collins, and he's going to be talking about cover crops. He is a master's student.
Can everyone hear okay? Sure. All right. And as Elena said, I'm going to be talking about cover cropping and really improving our sustainable weed management implications in soybean agro ecosystems. My name is Ryan Collins, and I'm advisor in the direction of Dr. Aaron Hiramoto. All right. So why an integrated weed management program? Well, I am a farmer, and a lot of things go wrong on the farm, and every tool I have in my toolbox makes me be able to solve problems a lot better. So a wrench doesn't solve all problems. Sometimes I need a hammer. Sometimes I need a screwdriver. And it's the same way with our strategy to manage weeds. An integrated weed management strategy with a focus on ecology is going to lead to a more sustainable model for future farmers. Implementing an additional management strategy can also reduce resistant populations, which is becoming the largest problem for weed science and farmers that are reducing yields, increasing costs for management. So looking at the ecology behind all of this, we can hopefully provide farmers with a more ecological, sustainable model for weed management programs in the future. In addition to this problem, no new herbicide technology has been developed in the last 20 years, and this is creating even more limitations for farmers. As we can see, over time, since the introduction of herbicides here in the late 70s, over time we have seen a large increase in unique populations of resistant cases. Moving on, we can see that further complicating this issue, there's also a rise in resistance to multiple sites of action. And sites of action are essentially just where exactly in the plant that these resistance processes are taking place. And you can see that already there's over 100 different weed species that have developed multiple sites of action resistance. And based on the table from 2008, we're already seeing some weed species that are evolved resistance to up to 11 different sites of action. So why mare's tail, also known as horseweed? Scientific name is Coniza canadensis. As of 2016, Weed Sciences Society has labeled mare's tail as the #1 most problematic weed in soybean systems. So why is this? Well, the life cycle is really complex. If you're a farmer in the South, you're going to see these populations of mare's tail flushing right after seed rain in the fall. And as you move further north, mare's tail turns into a spring annual weed. And luckily, where we're at here in Kentucky, we're fortunate enough to have mare's tail be a problematic weed most of the year. So through this transition period, as climate change, Kentucky, Kansas, these areas, Missouri, we're starting to see a lot different patterns in the emergence of mare's tail. And also, mare's tail populations exist already that with 5 different sites of action. When I say this, I mean individual sites of action, and there have been 6 different cases in the United States and Canada where there have been more than one site of action resistance in the population. Building on that, we can look at where mare's tail ranks with some of the most problematic weeds in terms of resistant to different sites of action. A lot of your turf grasses are here at the top of the list due to overuse of herbicides on golf courses. But some of our agricultural weeds are starting to make the list. You can see Palmer amaranth and water hemp as well here. And mare's tail has definitely made the list. And once they have the potential for having a resistance to multiple sites of action, we anticipate that these numbers will continue to grow. So we're talking about tools to the toolbox. So cover crops can effectively be one of these tools. And so they have many benefits for farmers. Those include holding on to nitrate, preventing it from getting into our water systems. We can reduce erosion, increase water holding capacity. But the main reason we're looking at cover crops is for weed suppression. And as you can see as we work across, the first photo here is an early season established cover crop. You can still see the corn stubble on the ground. Not as great a weed suppression as you work your way across, but you can still see that there are some biomass out there. It's taking up those nutrients that weeds could be feeding on. And then as we move across, that we see a spring herbicide or spring cover crop emergence or establishment, and the biomass has increased dramatically. This is before we terminate. And then after termination, we can see the soybeans coming through, but we still see that cover crop biomass providing a layer over the soil surface that is preventing light from getting to the surface, and therefore keeping some of these weeds that are cued by light from emergence. So what are our objectives? Looking to determine the impact of our diverse weed management practices, looking at mainly how do these affect marrow cell emergence, the development of marrow cell, and the growth. Secondly, we're going to characterize the marrow cell biology so we can better give farmers a model, and do that based on studying the weed. Once we know the emergence patterns, what makes the weeds emerge, we can then adapt these cover crops to hopefully better manage the species. And then we're going to look at the economics. Today we're going to look at some of the yield data, but in the plan, in the future, we want to provide a plan that gives an economic incentive for each of these management strategies, and then be able to compare them based on your site or location if it's different regionally. But like I said, today we're going to focus on the first objective for time reasons. Our experimental design, we've got 4 locations. I'm going to be giving data today from 3 of those, Kentucky, Kansas, and Illinois. We all conducted the same experiment in a no-till cornfield, or a no-till soybean field, and that's going to be following a corn system. Our plots are 3 by 10 meters. They're going to be arranged in a randomized complete block design, 4 replicants, and 10 different treatments. Here we're looking at some of our management strategies. We start off with at the top with an untreated check. This means no strategy whatsoever. As we work down, you can see we have a difference in overwinter and winter kill cover crops. We have a fall herbicide program. We have those with or without a residual herbicide. A residual herbicide is just a herbicide that stays in the soil a little longer, gives us a little longer suppression for weeds. We also implemented a fall tillage experiment or treatment and the literature tells us that tillage can suppress marrow cell a little better. And looking at the bottom one here, we have a weed-free management strategy where we use a multiple herbicide approach. All right, something about the soybean management. Our row spacing, 38 centimeters at 373,000 seeds per hectare. Our variety is a Pioneer 37 T09L. The L stands for Liberty Link technology, where you can come over the top with glufosinate. Our cover crops are going to be terminated with a Roundup PM, and you can see our termination and planting dates for the 3 sites that we're discussing as well. So our data collection, we have 2 small quadrants that are set up in each plot where mare's tail emergence is counted. We're pulling those to make sure there's no recounting. Yield data was collected and adjusted to 13% moisture. Weeds were rated prior to each herbicide application, and density and biomass were collected as prior to soybean harvest. And then cover crop biomass measurements are also collected prior to a herbicide application. Today we're going to be discussing a little more on the top 2. So on with our analysis, mare's tail seedling emergence is going to be summed over 2 different periods. We have a prior to soybean planting and a post-planting or before harvest. Yield data is going to be presented a little bit differently, analyzed the same, but we saw some different outcomes in the Illinois treatments compared to the Kentucky and Kansas treatments. So we're going to look at those a little bit separate. And all data is analyzed using SAS 9.4 with ANOVA, using state and treatment as 6 factors. So a little bit about our results. First, we're going to start off with the untreated versus treated, our untreated versus our rye-only treatment. And we can see by these that there is near a 2-fold significant difference in suppression in both prior and post-planting. Going on to our no herbicide versus any herbicide treatments, we can see a little bit of the same. There's a 2-fold significant difference. Some interesting notes that I found with this, when we added a residual herbicide to our herbicide program in the fall, we saw a significant difference in suppression of mare's tail. This, to me, tells us a lot about our flush of maristelle in the fall. So when our burn down in the early doesn't necessarily time up with the emergence of this maristelle population. And we can see that a little bit in the next comparison as well, where no fall herbicide versus with a fall herbicide, we saw basically no statistical difference. And this being said, I believe that the maristelle population is being missed by this early management strategy. Comparing the results, Kentucky and Kansas, as we noted, between treatments 2 through 10, as long as we had a management strategy, we saw no significant differences in yields, but those are significant compared to the untreated check. Yields were a lot different from Kentucky and Kansas. We had significantly higher yields, which you can relate that probably to the climate and geography. We get a lot more rain here in Kentucky, and that may be the limiting factor for Kansas's results. Looking at the Illinois results, as we said, we saw some significant differences, but unfortunately, they're not easily explained. You did see the highest yields in our weed-free plots, but as you see, there are some significant some yields that are significantly similar, but there's no really relation to a lot of those. And hopefully, our second-year data may be able to tighten some of these loose ends up. So our conclusions: treatments with a growth regulator and a residual herbicide saw significant suppression compared to the treatments without the growth regulator. Again, that was the residual staying in there a little longer, maybe hitting that timing of the flush a little better. In Kentucky and Kansas, as long as there was a management strategy applied, we saw no significant differences in yields. Again, so I don't know if we were just good farmers or what, but it's not really mattering what strategy we do implement on our yields. And finally, treatments using a cover crop or herbicide saw a significant reduction in maristelle emergence compared to the untreated check. And again, when we say significant differences, these are not in terms of economics. These are just in terms of differences in the weight in the yields. Further research, we're going to still build on quantifying the timing and the frequency of maristelle biology across the transition zone. We're going to do that by utilizing soil moisture and temperature to predict these emergent maristelle populations, and hopefully we can give people outside of our geographic locations some guidelines for this as well. And then thirdly, I'm going to be working with Dr. Jordan Shockley, who's on my committee, and we're attempting to build an economic model that places a value on each of these treatments, and then we can provide a return on investments to farmers, because at the end of the day, farmers care about the money. If it's not going to work for them, save them money, they're probably not going to do it. So, and with that being said, I'd like to acknowledge my funding, my committee, collaborators, and the Hormoto Lab for all the help that they've given me so far. And I'll take any questions.
Do you have any questions? I wanted to know, so how would no-till system suppress the maristelle? What's the primary way that they suppress it with cover crop?
Well, we can answer that in many different ways, but the main reason the cover crops are suppressing is just by outcompeting the weeds. And maristelle germinates on light cues as well, so any biomass that we can have covering the soil surface is going to limit, and it's going to suppress those flushes of maristelle populations emergence.
Thank you, Ryan.
Thank you.
Thank you. Okay. Yeah. All right, now we're going to hear from our next speaker, Virginia Verges, and she's going to be talking about wheat, and she is a PhD candidate.
Thank you, Alaina, for your presentation. I'm going to talk today, show a little bit of my data from my PhD and working with Dave Van Sanford in wheat breeding. And first of all, to start, I'm going to say a little bit what is genomic selection. That is what the main, applying genomic selection is the main topic of my PhD project. So genomic selection is a plant breeding tool that helps modeling to predict how the performance of a genotype will be without field testing, and how genomic selection works. So basically the main objective, the central objective of genomic selection is to calculate the estimated genomic breeding value. And this is done for selection candidates that have only genotypic information. And how we do that? We do it building a training population with lines that have both genotypic and phenotypic data. So with this training population, we train a model, and that model will calculate the estimated genomic breeding values. And with this, the breeder will be able to make selections and keep the lines that wants for the next cycle of breeding. So how we measure the success of this model, of the genomic selection model we are using? We measure it with calculating the prediction accuracy. The prediction accuracy is the correlation between the phenotypic real value and the predictive value for each line. And what we want is to maximize the prediction accuracy to be sure we are having good predictions. There are many factors that affect the prediction accuracy. Some of them are shown here. The training population size, for example, the number of markers used, also the statistical model used, and the relationship between the training and the validating population. And so far, this tool, genomic selection, has been used in the last 10 years, or started to be discussed in the literature and for actual breeding programs in the last maybe 10 years started, and has been is showing some advantages. So that's why it's very interesting for breeders. Why is, the first one is that it shows an increase in the selection gain per unit time by reducing the length of the breeding cycles. Another very important advantage is that this model can capture minor effect QTLs on markers that are spread on the whole target genome. And on the other hand, has shown also to be very useful in assisting for selecting parents for crossing. So why we are talking about using genomic predictions in preliminary trials? Preliminary lines, as you can see here, preliminary lines is the first year of a field evaluation in a breeding program. So the breeder has many lines that has to evaluate in the field, and generally doesn't have much seed. You have few seed, maybe for 1 or 2 plots. So it would be really good to be able to have predictors, to have breeding values for these lines, and maybe avoiding some field testing. Or maybe not avoiding the field testing, but adding another very important source of information to do the selections. So with all this said, the main objective of my project is to design an optimized genomic selection for preliminary lines in the UK with breeding program. And in particular, we will try today, I will show how to evaluate the model predictive ability for different populations, and also to compare the prediction accuracy with different training population and genomic selection schemes for different traits like yield, test weight, heading date, and height. So to get this done, what we did was to select 6 sets of data to build 6 populations coming all for the wheat breeding program. Sets 1 to 3 are multi-environment replicated sets, lines that were planted in 4 locations with 3 reps in 2 years, and set 4 to 6 that are actually preliminary lines were planted at 2 locations in 1 year last year. So the data collection was done during 17 and 18, 2 years, and at 4 locations in Kentucky. And we collect grain yield data, test weight, heading date, and height in all the plots in all locations. The genotyping was done in the lab, in the genotyping lab at North Carolina State. We sent samples of leaves there, and they did the genotyping, inputting the markers, filtering, and finally we got a final hub map with 2,400 SNPs. This for every line, no? That is part of the project. So finally, how we run the predictive model. If we go to this previous scheme, the genomic selection scheme, what we need to accomplish this, as this was saying, we will have first to define a training population, define the selection candidates, a validating population. Also to define the prediction model, we use RRBLAP and we run it in R. That was the software to train the genomic selection model. And with this, we could test the model predictive stability with cross-validation and apply genomic selection with a training population and validating population being completely independent sets, and also applying genomic selection, but with but what we did was to split a set in 50% going to the training population and 50% being the validating population. So we predict half of the families with the other half. So to start looking at some results, these box plots are the mean yields, the mean and the deviation for the four traits that we investigate. We have here means for grain yield, heading date, test weight, and height. And it's interesting to see the year effect as we see the 3 sets, 5, 4 to 6, that were only planted in 2018, had an average lower yield compared to the sets with 2 years of data, and also had a later heading maturity as an average of the 3 other sets, and also a little bit lower height and kind of similar test weight. So this is the prediction accuracy. Let's remember the prediction accuracy is the correlation between the predictive reading values and the real field information. So that's the measure of how our model is working. So here we have prediction accuracy for the 4 traits for the 6 sets individually. We applied a cross-validation sampling scheme where the cross-validation was run 100 times, and each time the model was picking up 80% of the lines to be the training population and 20% of the lines to be the validating population. And with this approach, we got accuracies that we can see here from 0.32, all moderate to good, moderate to strong, 0.3 to 0.6 for yield, 0.4 to 0.7 for test weight, 0.3 to 0.7 heading, and 0.3 to 0.7 for height. So this will show that the model was working well in predicting the breeding values. And next, what we did was to predict the preliminary lines that are set 4, 5, and 6 with training populations that were completely independent sets, lines in sets 1, 2, and 3. So we have here like a factorial combination for the 4 traits again, and we got a lot lower accuracies. Some even negative, and we have for the 4 traits, the maximum here 0.3 for yield, for heading date a little bit better, 0.5, for height 0.3, and for test weight also 0.3. So we got with this approach lower prediction accuracies. And then what we did was the other selection scheme to split the set 50%, each 50% in the training population, 50% in the validating population. And with this approach, we applied this now for the preliminary lines. And with this approach, we improve a lot the prediction accuracies. We have for yield, for example, ranging from 0.1 to 0.6, test weight 0.4 to 0.6, heading date also all moderate to good, strong prediction accuracies in height from 0.3 to 0.5. So this is a very good improvement. So what does all these numbers mean for a breeder, for the breeder perspective? And we wanted to analyze this, especially for yield, not only because it's a very important trait always, also because the field testing is expensive. And one of the reasons genomic selection is becoming so popular is can save a little bit of money, because genotyping today is becoming a lot cheaper, or is becoming much cheaper to genotype a line. So it could be a good strategy to maybe save your resources for later stages in the breeding program. So with this said, I'm going to show the 3 scenarios with the accuracy we had. For example, set 5 for a prediction accuracy of 0.6. And here we have plotted all the lines, the actual phenotypic yields here in the x-axis, and the predicted breeding values in the y-axis. So we can see what could happen if a breeder uses a 20% selection intensity that is pretty average, will select the lines from this line to the right. And if the breeder makes his selections or her selections based on the breeding values, will make it from based on the predicted breeding values, will choose the lines from here to the top. So what we can see here is that if we see where are the lines that were chosen by the 2 strategies, we can see that 13 of 16 lines were captured, both selecting based on phenotypic selection and also based on the predictive values. We can see the same with a prediction accuracy of 0.4. It's also good to see here in this area the lines that will be predicted based on the field data and based on the breeding values. And it's also good news for the breeder. Here we can see that 10 of the 14 lines will be captured in both ways. And finally, we have the worst scenario, when we had a prediction accuracy of 0.13. Here we see that only a few lines are chosen by both strategies. Only 3 of the 14 lines were captured, and this is not good news. Sometimes this also can happen. There are some populations, so there could be reasons that we need to investigate how this could happen with some genotypes. So finally, as some conclusion from my project, the evaluation with cross-validation show moderate to strong prediction accuracies, and predicting sets with independent sets of data show low to moderate prediction accuracies. Yes, low to moderate, sorry, prediction accuracies. And when we set up the scheme to predict 50% of the lines with the other 50% of the set, we improve a lot the prediction accuracies. For grain yield, this improvement meant a 30% to a 300%. And for an important trait like yield or training prediction accuracies like 0.49 or more, gives the breeder an excellent tool for selecting lines based on their genotypic value without field testing. So with all this said, thank you very much to all these people, and I'm open for any questions you might have. Well, the model that we use I think the model we are using in RR-BLAP, when you have is actually like the script to run it, and we try with these 4 traits, yield, test weight, heading date, and height, to try to evaluate if the model was working good or not. It's a model that is pretty used right now for many other breeders or breeding programs. Maybe some other models could include covariates, some other like strategies. And what we did was to train this model with our 4 traits to see if it was good or not to be used in this type of research.
Thank you very much. Our next presenter is Sanjay Joshi, and he's going to be talking to us about Arabidopsis embryogenesis, and he is a PhD student.
Hello. Thank you, everyone. I'm pleased to introduce my research. We belong to seed biology group. We work on the lab focuses mainly on the gene regulatory networks, which are involved in the seed embryogenesis. And today, here I'm going to present one of my projects, which is the role of transcription factor LAB40 in Arabidopsis embryogenesis. Before I begin my presentation, I would like to ask a question to you, that how much % of our food diet consists of seeds? Because we are dealing with the seeds every time. I will answer this question during my talk. I would start my talk with, with introduction of somatic embryos. We work with the somatic embryos. These are the artificial process to produce the embryos from the somatic cells or the vegetative cells. You might be wondering what is the importance of the somatic cells. It looks like the zygotic embryos, but it is made from the somatic cells. And it is important because it helps to regenerate the transgenic plants to meet the food demand. As our population is increasing, the food demand is been increasing day by day. Or to test the genes that we do in the lab, we test using the somatic embryos. However, in plants, it is still inefficient. It is not easy to get the somatic embryos. So it makes us a challenge to understand the system. And the question that I ask, how much % of the seeds that we use, is like 70% of our food diet consists of seeds, which makes really significant importance to understand the seed biology, to understand its gene regulation. Apart from that, somatic embryos help to get easily accessible compared to the zygotic embryos, which are embedded in the maternal tissue. So somatic embryos are easy to do in the lab work, and it takes shorter time to study, and it gives the same result that we expect from the zygotic embryos. So I would now share what is known in this somatic embryogenesis. One of the key transcription factor, agamas like 15 or AGL15. When I say transcription factor, the transcription factors are the proteins which bind the DNA and regulate the gene expression. So they have a key role to binding to a DNA region which are specific, and they either increase the gene expression or decrease. So they turn on the gene or turn off the gene. So one important transcription factor that well studied and well known, and in our lab we are working, is agamas like 15, that's AGL15. This is heart-shaped embryos where you see the brown are the staining of accumulation of AGL15. And apart from that, AGL15 promotes the somatic embryogenesis, but also is involved in the flowering phases. And AGL15, this is the complex like gene regulatory networks, where AGL15 is in center here, and it directly increases, regulates, upregulates some gene or represses some gene. However, the relation between AGL15 and LAB40, which I will talk next, is not yet known. So what is LAB40? So LAB40 is also another transcription factor, which means it will bind to DNA and it will help to either turn on or off the gene, which is belonging to another family, which is lateral organ boundary. From that, it got the name lob. So it's a lob domain family, unique to the plants. And it is present in mostly in the seed development. This is the developmental plant development map, heat map. The yellow color shows the low expression of the gene, and the red color shows the high. And it is very clear that we see on the seed development, the accumulation of this protein is high. But specifically in seed, it's present in the embryo, which makes it more interesting to study what is its role. And previous literature studies which have been done, and they have shown that LAB40 is expressed highly in other development, other embryo-related genes, which are LEG2, FUSCA3, and ABI3. These 3 genes are also important for embryo development. And these all have shown that LAB40 is expressed in good amount, which makes us a good candidate to study. So before I came in our lab, there was already East2 hybrid system where they studied, and they showed AGL15, which promotes somatic embryos, and LAB40, they interact. With this assumption, we hypothesized that LAB40 has a critical role in somatic embryogenesis. And the main objective of my study is to know the overall embryogenesis in Arabidopsis. But specifically, I am working on to show the interaction between LAB40 and AGL15 in plant, and to know where the LAB40 binds in DNA. We don't know yet where it binds directly and what it is doing it. To do so, I'm working with the Arabidopsis plant, which is a model plant. It's short and easy to grow. And from Arabidopsis, I work on the seeds, because we are focused on the seed, and this embryo culture tissue, which are subculture in the lab. So these are 2 main plant materials that I work. And now I want to show first result. Before illustrating my result, I would give a hypothetical example that what is co-immunoprecipitation. Here I'm trying to show that AGL15 and LAB40 interact. So those 2 proteins interact with each other. And this is done with one method, co-immunoprecipitation. So when you have like 2 proteins, say X and Y, and you think that they are working together and they're always together. So to do that, first you will immunoprecipitate using one antibody, say X antibody, and then you will do the blot with the Y antibody to see. If they are together, you will find a band. If they're not together, you will not get the band. But in my case, they are working together. So what I did, I took the tissue of LAB40, grinded them, and did the immunoprecipitate using AGL15. So this is another antibody. And then when I did the Western blot, I used anti-MIC, because my LAB40 has anti-MIC tag in it to detect. So when there is the anti-AGL15, I see a band. And when there is no anti-AGL15, I don't see a band, suggesting that LAB40 and AGL15 are interacting with each other. One of the important phenotype that I found while working the embryo related is from the SAMSE experiment, which stands for shoot apical meristem somatic embryos. In this system, what we do, we take different lines of seeds and we put it in the media with 2,4-D, and after 21 days, we count the embryos. And we see how many seedlings have the embryos and how many they don't have. And this experiment, this Columbia is the wild type, that is the control. And we see like 25% of the seedlings had embryo. And the overexpression line of AGL15 is always having a high amount of embryos. To see here, when I took the loss of function, that means when there is no LAB40, and the 41 is the closest homologue. So I took LAB40 and 41 line without the gene was mutated, and found that there is significant reduction in the embryos, suggesting that LAB40 and 41 has an important role in to give more embryos. And these are the pictures, how the seedlings look like when they pop out the embryos. And so this was one of the important phenotype that we found and encouraged us to see, okay, if this gene is really related to the embryos, let's figure it out where in the gene regulatory network, where it binds. So for that, the experiment we need to do is chromatin immunoprecipitation, also called CHIP. Here I would like to talk briefly about what is the protocol. It's like 3 days protocol where you have a protein of interest that has a tag. The first day you glue the tissue with formaldehyde, which helps to fix your protein and DNA as it's embedded in the plant. And now you grind the tissue, and here you see the purple color are like of my interest, and there are other protein that I don't want. So for that, what I do, I grind the tissue and do the sonication first to shear my DNA, to chop the DNA, and put the antibody. Antibody of my interest, I will put anti-MIC tag, and after antibody and saffrose beads, the protein will be immunoprecipitated. And then the next step will be to reverse the cross-link using the heat and do the PCR, because we want to know where in DNA this protein binds. So this is one of my results that I'm showing. this is the Western blot to convince myself the protein that I'm working is still working in each steps. So whatever steps I work, I want to confirm myself that this protein is there. And in this, you see the post binding, when there is protein, there is depletion. When I put the antibody, there is depletion of the protein. And here, when you see when there is plus, there is protein, and when there is no antibody, there is no protein, suggesting that the system is working. But I don't know how or where it binds, right? To know that, we need to do sequencing. So we send it for chip sequencing, and when the sequencing result will come, we will know its potential binding site. For now, I don't have the binding results yet, but soon I will get within this week, I think. But another step is to know the transcription level, which means if this protein is working and it's helped to regulate the gene, it means it is turning on or turning off some genes. We need to know which genes it is doing. And then while combining the chip result and RNA-seq result, we will know the direct and indirect targets of Lab40. So I did one RNA-seq result with the help of, in the developing seeds, which were 7 to 8 days old seeds. So I collected the RNA and then did the sequencing and found that 335 genes are expressed and 32 genes were repressed in that developing seeds. And when I use those list of genes and then put it in this Panther software, which gives the gene ontology enrichment test. So it categorizes the genes, whatever you have found, based on their functions, biological functions. And this is a part of this graph is part of my full result. Here I have chosen the biological process based on which previous literature review have shown, which has an important role in somatic embryos. So lipid localization, response to water depletion, response to abscisic acid, response to hormone, and response to stress. These are significantly enriched in the overrepresented data, suggesting that the genes which I have found, all the 335, among them, some genes have crucial role in embryogenesis. So as I discussed my project, some of the literature work that has already been done, like AGL15 binding with FUSCA3, LEG2, ABI3, which are crucial embryo identity genes, but we don't know what is happening between Lab40 and which are the targets of Lab40. And if we know this, we can predict the gene regulatory networks and find out the potential things that we can directly help in developing soybean seeds or other seeds, the plants which have difficulty in even getting zygotic embryos. So it will be a breakthrough. So the future work that I want to show is like I will do another RNA-seq experiment using the mutant line and the overexpression line with the SAMSC that I discussed before and compare the results of SAMSC and the seeds that are developing that I already did. So this will give us an idea how many genes are common in the developing seeds that I did and the SAMSC. And furthermore, I need to validate my ChIP-seq result data by doing qPCR and repeat the ChIP-seq again. And another thing is I need to still confirm the interaction between AGL15 and Lab40. This I will do with crossing different lines of the plants and to know what is happening between them. And the next work I need to find is in the DNA, where does it bind to be exact? That is the cis motif. To know that, I need to do the electrophoretic mobility shift assay that helps to know us that in the DNA what genome sequence does Lab40 binds to. With this, I would like to thank my professor, advising committee, my lab mates, my wife, and thank you for everyone listening. If you have any questions, please. Yes, please. Yes. Yes. Soybean. Yes. No. This is like the model plan that we are studying now to speculate. So we have to, if we have to see in monocot, we have to do other experiments. to know it in better. Okay. Thank you. Thank you.
Our next presenter is Bryan Brady, excuse me, and we'll be hearing some more about wheat, but in a different way.
Okay, so yeah, we're going to be hearing about wheat in quite a different way with this project. So noticing that this is a little bit of a wordy title, we'll work our way through it. Building a better baguette. So volatile organic compounds, sensory analysis, and baking quality assessment of artisan bread made from modern wheat varieties and land races grown in Kentucky. So a little bit of background. Demand in locally produced grains has increased in recent years. And this is among small-scale millers, artisan bread bakers, pastry chefs, local food retailers, consumers, kind of across the value chain board. And so this provides an opportunity for small-scale farmers to kind of get into this value chain. When you can access a local value chain, a lot of times you can cut a couple middlemen out, make a little bit more money per acre. Get a higher percentage of the food dollar. And for some farmers that are already growing wheat, this may give them the potential to add another cash crop to the rotation. So what's radical about bread wheat in Kentucky? Well, geographically, Kentucky is a soft winter wheat region. And culturally, we're more of a biscuit state than we are a bread state. So that comes from the lower protein flours that we produce. The lower the protein, generally the lower the gluten, the more tender the product. But that being said, local bread bakers in Kentucky want to create this niche value chain for hard winter wheat, which is more suitable for artisan bread products. And when we say artisan bread products, what I'm talking about is lean doughs, just flour, water, salt, and some form of yeast. No shorteners, no softeners, anything like that. And so out of this demand from local bakers, the Baguette Project was born. And this demand for local hard winter wheat is actually not unique to Kentucky. There are areas really all over the United States, but some of the areas where this is concentrated in is like Asheville, North Carolina, central and upstate New York, Vermont, Maine, especially in Washington State, where Washington State University has a bread lab. So it's not unique at all. This is something that's kind of happening across the country. So modern varieties of hard winter wheat are being evaluated and bred to produce acceptable yields with our high humidity and high rainfall. High yields and high protein content. Obviously, that's important. We'll get into that a little bit more. And so this project is tasked with the evaluation of these hard winter wheat varieties and land-raised varieties in regard to management, yield, baking quality, and sensory experience. And just a note, the reason that we brought land-raised varieties into this, there was a First Friday talk a while ago where there was kind of this idea that, there's this idea among bakers that some of these heirloom grains might taste better. And so that was something that we were interested in kind of evaluating, particularly if you're going to make white bread out of something, is there actually a difference in taste? We'll get into that a little bit later. But these are the 4 modern varieties that have been bred for the Southeast. And then these are 4 pretty much land-raised varieties that Dr. Van Sanford found in an old USDA Ag Bulletin from 1920. These are 5-foot-tall pre-hybrid varieties. They look gorgeous in the field, but that's about all they're good for. So we had 2 management types. Another thing that was kind of brought up with some of these heritage varieties is that if you manage them in an old-fashioned way, let's just say fall manure, no spring shot of nitrogen, that maybe they would produce better. We thought, well, let's just go ahead and test that. So those are the two nitrogen management types. One is just a typical fall nitrogen, and then spring shot of nitrogen. And so we'll call that the conventional management type, and then we'll call the other one a heritage management type. And so right off the bat, just a look at yields, conventional management, we can see right away that the bottom four are the heirloom varieties, and we expected that. And we can see that they still are the bottom four in heritage management as well. And so the overall across the board mean reduction in yield between the conventional and heritage management varieties was 15.7%. But you know, talking about that heritage management and how that might help the heirloom varieties, we actually saw an even larger reduction in yield between the conventional and heritage varieties, between the conventional and heritage managements with the old landrace varieties. So even bigger reduction in yield, 20.9%. One thing that we did notice that might that might have you know, a benefit of using those old heritage techniques, or really just fall nitrogen, is that the lodging percentages were double in the landrace varieties under conventional management. And that's really from that spring shot of nitrogen. You get more biomass, they just weigh more, they're 5 feet tall, they blow over. There was no significant difference between the two management styles in flower protein, and that was relatively shocking. And then a comparison of the means, you know, again, shows that all the landrace varieties yielded significantly less than all the modern varieties. And so another category that we're evaluating, baking quality. What does that mean to evaluate baking quality? From an artisan baking perspective, we're looking at gluten potential. That really is the building block of good bread. The flour protein doesn't really tell the whole story. All of these flours from these varieties were in the 10 to 11% range. That is relatively low for making bread. You know, gluten is a product of water and flour, and when you add water to flour, the two proteins, gliadin and glutenin, gliadin, which is responsible for extensibility in dough, glutenin, which is responsible for elasticity, they get together and they form a gluten network in the dough. And glutenin is really perhaps the most important factor in artisan baking quality. That's the ability of the bread to hold its shape, of the bread to hold the gases in as fermentation happens, and then also as the bread to hold itself together in that same shape when it's baked. Loaf height is another thing that we are going to use to evaluate baking quality. Oven spring is basically when the loaf goes into the oven, it's going to expand like a balloon, but at some point that extensibility meets elasticity. There's a little bit of constriction back, and then that bread sets. But you do want it to be able to expand. You just, like a balloon, you don't want it to pop. When we talk about crumb in this case, from a white flour artisan bread perspective, you are looking for open crumb, light, airy loaf. And you also want that crumb to be evenly distributed, no dense spots. So this is what we call a baker's cut. It's kind of a horizontal cut, and right away you'll notice these are shorter than most baguettes, and that's really just a product of the depth of the oven that we were able to bake in. So these are pretty much demi baguettes. And so when we look at the crumb right here, you can see that these two in particular are quite a bit more open and have some of these larger air pockets in here. A little bit hard to discern, you know, if the crumb is better from this horizontal cut, but we've got a better picture, I think, that really, let's advance. I think this tells the story a little bit better. These are cross cuts. And again, here, the New East variety and the Virginia variety are clearly better. You can see a lot more density in this variety And these are, you'll notice that there's been 2 heirloom varieties that have been kicked out. That's because their yields were so low we couldn't even get enough flour out of them to make bread. So Fulchester and Fultz are the only heirloom varieties left. You can see that they're a little bit more dense, especially Fultz. And again, we were talking about low fight. You can see how this low fight compares to this low fight. So we'll go ahead, and this is another way to look at it. This is kind of like a crumb print. And again, I think this shows, you know, more evenly distributed large holes. Another thing that we can use to, another measurement that we can use to look into gluten potential is lactic acid solvent retention capacity. And you can see those varieties that had the better crumb had the higher lactic acid solvent retention capacity, and that indicates more gluten potential. And you can see that low fight is relatively the same order. So we've got 2 clear baking quality favorites. So the next category is sensory experience, and bread really is a sensory experience. You use all of your senses with bread. You hear the crunch, you know, you smell bread. There's nothing like, you know, when I cut open bread in the lab to do stuff, everybody turns their head and it's like, oh, what's that? This is my research, not lunch. So from a sensory experience perspective, we're looking at crumb, texture, aroma, taste. Again, the more open, the better. That is a greater crust to crumb ratio. The crust is really where it's at with bread. I mean, don't tell kids that, but as far as baguette goes, that's where all the Maillard reaction stuff happens. That's where the caramelization is. So the more crust you have to crumb ratio, it kind of ends up having a better effect on taste. Texture, we're looking for crispy, chewy, and creamy, while also seeming light. Aroma, again, aroma has the greatest influence on flavor. We'll talk a little bit more about volatile organic compounds here in a second. Taste is really influenced by all the above. So these are our semi-trained sensory panel results, and unfortunately you can see there's really there's really no significant difference, and we sort of expected that with white bread. So, you know, the variability is just too large. When you do a taste test with a Likert scale, somebody's 2 is different from somebody's 5. There are other ways to look at this. We may end up going and doing triangle tests where we put 2 of the same with 1 different and see if people can at least discern those differences. But these flowers have different carotenoid pigments, so we'd have to blindfold you. And I'm not sure I want to deal with IRB and blindfolding people, but we'll see. So another way to kind of take a look at these volatile organic compounds is, these volatile organic compounds are so responsible for flavor that I think all of us have had a cold before and have not been able to taste. Your tongue, you know, can indicate that something's salty, something's acidic, something's sweet, but you can't really taste what something, you know, that actual taste is really part of this olfactory bulb right here. And so what we're doing, and this is the part that's we're still kind of in this process, is looking at the Headspace Analysis, basically using a process called solid phase microextraction, where we're going to take part of the crumb, we crush it up with liquid nitrogen, we put it in a vial, in a solution, we insert a fiber into that headspace, that fiber picks up those VOCs, then we put it in a GC, desorbs, and then we can basically take a look at any kind of quantifiable differences between VOCs by variety type. And so kind of reaching a conclusion here, you know, we've got the 6 varieties that we held on to over here on the left, and by category, you know, I basically used a green light, yellow light, red light visual indicator, and we can see that New East really seems to be the clear winner so far, and the VA09 hard red wheat variety also did really well. Even though we didn't have any significant differences with the tasters, we still had them rank, you know, rank the orders of the bread. So I ended up using those to kind of indicate. Not entirely sure why this one ended up doing so well that as far as the taste test, but it could definitely be the fact that it's just the better texture, the better crumb leads to the better taste experience. And so with that, I'll wrap it up, and definitely want to say thank you to all the people that have helped in this project, and I'll take any questions. Sure.
So, storage can decrease that over time. Really affect after steady
Sure. Yeah, and milling definitely affects the you know, the enzymes in bread flour big time, especially depending on the temperature that milling. You know, the roller mills generally mill much cooler than stone mills, and so you know you can lose some enzymatic activity in flour, and you know some of that enzymatic activity is something that a baker wants. You know, really like some amylases in there to break down into more simple sugars, make the bread taste a little bit better. So yeah, I think there's definitely some research out there that suggests that milling techniques can greatly affect the end product of the bread.
Now we'll conclude this section, so we're going to lunch.
Yep. Hopefully lunch is served. So we'll see everyone back here